Okay, so hi everyone. My name is Chelisa from the Translational Research Lab over at the Canberra Hospital. So as Matthew mentioned, I have just started my PhD this year, and I did do an honours year last year, and that was a time in which I dedicated most of my work towards this project that I'll be talking to you about today. So my project focuses mainly on a particular mutation that we believe leads to the development of common variable immunodeficiency. So first of all, what is common variable immunodeficiency? So otherwise known as CVID. It's a disorder in which your immune system is considered to be impaired or weakened. So the normal function of the immune system is to prevent the occurrence of infections and diseases by fighting against foreign invaders such as bacteria and viruses. So with patients with CVID, this function of the immune system is weakened or defective. Some of the characteristics that can be present in patients with CVID are low immunoglobulin levels. So immunoglobulins are otherwise known as antibodies that are produced by white blood cells to recognize and bind to bacteria and to be able to aid in the destruction of those foreign invaders. So typically in CVID patients, these immunoglobulins are reduced. And these immunoglobulins are found to be more so reduced in IgG, IgM, and IgA. Patients with CVID also tend to have high susceptibility to infections due to that weakened immune system. These are mainly identified in your lungs, sinuses, and your ears. There is also the occurrence of autoimmune diseases, which are found in about 20 to 25% of CVID patients. Now, there are a wide range of autoimmune diseases that are found to be linked with CVID, and these diseases generally occur as a result of your immune system actually developing an inappropriate response against your own cells, meaning that your immune system begins attacking your own healthy cells. Examples of such diseases include rheumatoid arthritis and alopecia. Granulomatous diseases are also identified. About 10 to 22% of CVID patients have this disease, and these occur as a result of the development of granulomas, which are basically masses of immune cells that are present at sites of infection or inflammation. And finally, neoplasias, which are most commonly known as tumours, are also identified in some CVID patients. So a difficulty with CVID patients is that they can be asymptomatic, meaning that they don't show symptoms. So this can be quite difficult in determining the exact prevalence of CVID, as it varies quite greatly, especially in different countries. However, the estimated global incidence of CVID is considered to be approximately 1 in 25,000 people. This particular disease also affects both genders equally and is known to be present in later in childhood years or early to mid-adulthood. Now, in regards to the diagnosis of CVID, there are generally three set criteria that are followed. So the first is the presence of hypogammaglobulinemia, which is characterized by the reduced immunoglobulin levels that I mentioned before, again typically identified as low IgG, IgA, or IgM. There is also the presence of recurrent infections, as I mentioned, and the most common type is sinopulmonary infections affecting the lungs and your sinuses. And there's also the presence of impaired functional antibody responses. So as I mentioned before, antibodies are important in recognizing bacteria and being able to aid in their destruction to prevent infections and diseases. So CVID patients tend to have defective antibody responses, and these are generally tested in patients based on their response to vaccines such as diphtheria and tetanus. So if we currently have the ability to diagnose CVID and we know some of the characteristics that are present in CVID patients, then what remains a problem with this disease? Well, the primary cause of CVID still remains unknown despite about 40 years of research. Part of the problem is the heterogeneous nature of CVID. And what I mean by that is that this disease in patients can be caused by many different genes. So genes make up basically who we are. And genes in CVID diseases are known to be defective and mutated in these patients. However, it remains our goal to be able to identify and investigate these genetic mutations to be able to understand the development of CVID and also understand the normal function of immune system and immune system regulation. Some of the mutations that have been identified in NF-kappa B, some of the mutations that have been identified in CVID, have been identified in an important immune system called NF-kappa B. So NF-kappa B is a regulator of genes involved in immune and inflammation responses. So it basically acts to help regulate the immune system to help prevent the occurrence of immunodeficiency and autoimmune diseases. It's considered to be the central mediator of the immune response. So how you can relate the role of NF-kappa B in the immune system is like the role of a referee in a soccer game. So a referee has to make sure that all the players in a game have to follow the rules. With NF-kappa B, it has to make sure that all the components of the immune system function normally. If a referee does not do its job, you end up with chaos in a game. If NF-kappa B does not do its job properly, then you can result in immunodeficiency and autoimmunity. Now, there are two forms of the NF-kappa B pathway. One is called the non-canonical and the other canonical pathway. My project focuses mainly on the canonical pathway, which is this pathway that you can see here on this slide. And it's a pathway that is present in most cells of the body. As you can see, the activation of NF-kappa B involves many different proteins. However, for my project, the protein of interest that we looked at was this particular protein called IKK beta. Now, before I go into a little bit more detail of my project, I want to just touch on the significance of this type of research. So as I mentioned before, being able to identify and investigate these mutations allow us to understand more so the development of CVID. But not only that, being able to investigate these mutations allow us to better understand and add to the knowledge gained about the immune system function. And also other immune system implications such as autoimmunity and cancer. This knowledge gained would then be quite beneficial for future patients living with these immune conditions as more effective diagnosis and treatment methods could be developed against them. So prior to my honours year, our lab had identified a novel mutation within a young patient in her DNA sequence. And this patient presented with recurrent infections, specifically cyanopulmonary infections, and also reduced immunoglobulins, which were the characteristics that I mentioned to you before. So her symptoms did show signs of the development of an immunodeficient disorder. So from this discovery, our lab wanted to determine, I might add that this mutation was identified in the IKK beta protein that I mentioned to you before in the NF-kappa B pathway. So from this discovery, our lab wanted to determine the consequences of this mutation and its role in the possible development of CVID within this patient. And with that, we used two specific research techniques, Western blot and flow cytometry analysis. So Western blot basically allows us to detect protein expression levels of a protein of interest amongst a sea of other proteins. So it basically allows us to determine how much of a protein is present within an individual's cells. Ultimately, this technique allowed us to determine that the mutation in IKK beta resulted in a gain-of-function effect. This means that the function of IKK beta in the patient is considered to be more enhanced than normal when you compare to a healthy control. The next technique that we looked at was flow cytometry analysis, which is a laser-based technology that's used for cell counting. It allows us to look at specific immune cell populations and determine if the amount of cells within those populations are considered to be different to a normal patient. And how I would describe the concept of flow cytometry is kind of similar to visiting the supermarket. So when you go grocery shopping, you choose the items that you want, you take it to the cashier. The worker then scans the item one at a time with its barcode and identifies that item. This, once the item is then identified, sometimes the worker then groups similar items together, for example, fruit and vegetables in one bag and household items in another bag. Now imagine in your mind that this whole process is automated. Replace shopping with biological cells, and replace identifying items with a barcode with identifying cells with cellular markers. And that's what you have as flow cytometry. And similar to shopping, you receive all your items on a receipt, which basically represents as your results for your shopping experience. But with flow cytometry, you present your results such as these. Oh, sorry. These, you can see that different cell populations are separated from one another based on their size and also the cellular markers that they express, and these individual dots here represent an individual cell that falls within that cell population. So with this technology, we looked at specific immune cell populations within the patient and compared them to a healthy control. And what we identified was that there were these differences in CD4 positive, CD8 T cells, regulatory and memory T cells. There was reduced number of cells in the CD4 and CD8 in the patient compared to healthy, and also an increase in the regulatory T cells and memory T cell compartments. Now, to be able to prove causation from a single patient and to be able to justify that this mutation correlates to the CVID in one sample size is considered to be quite difficult. So with that, our goal of testing causation was done by generating an accurate mouse model through the CRISPR-Cas9 technology. This tool allows us, this genome editing tool allows us to generate a mouse model that has the same precise mutation that we've identified in the patient. And this tool enables us to edit DNA by taking and removing parts of it to therefore generate the same sequence as the patient. I might add that this isn't actually what we do in the lab. We don't take a scalpel and cut your DNA up. This is just a representation of what happens at the molecular level. So some of you may also ask, why mice? Well, mice are considered to be the experimental tool of choice for immunologists based on the fact that their immune systems are considered to be quite comparable to that of humans. So with this mouse model, we used similar techniques and looked at similar immune cell populations in the patient and the mice. And what we actually found was that there were the same immune system defects present in the patient and the mice. And this kind of gave us an idea that the mutation indeed in the patient was leading to that CVID phenotype. And the gain of function effect identified in the patient was also present in the mouse model. And since this gain of function effect in IKK beta has not yet been discovered, we believe this to be a novel finding. So in conclusion, my project revealed that the patient and the mice had similar immune system defects. And again, since this mutation has not yet been discovered, we believe that this CVID phenotype in the patient has led to the discovery of a novel human immune deficiency. This project has not only allowed us to add to the knowledge gain about development of CVID, but also normal immune system function and regulation. And hopefully these types of research will enable us to provide better diagnostic and treatment methods for patients with such diseases. And again, for my PhD, I'll be further analysing this mutation, not only looking at how, not only trying to test the causation of this mutation in CVID, but also to look at its specific mechanism within the immune system to better understand its role. So with that, I'd like to thank Matthew and also other members of the Translational Research Lab, members of the Centre for Personalised Immunology, as well as Gaetan from the John Curtin School of Medical Research, who actually engineered the mouse model that we used for this project. Also funding from NHMRC and also the Harvey research program. Thank you.